Is the audio video clear, people? Welcome back. So in the previous session, we had looked at the various principles and the tools. Okay, fine. Now we'll look at the details of the tools. कौन use करा जाता है and केमिति. This is a very interesting and important portion. So my request to all of you, join करो फटाफट. Otherwise you will lose it. It's both interesting and confusing also. ठीक अछि. Our concepts good are clear now, alire babu. Very difficult questions kariba. And believe me, in the NEET examination, it's two, तीनोटी रो चारोटी question compulsory ase. So conceptual clarity is very, very crucial. So फटाफट join करो quickly. Yes, जल्दी join. आ जाओ बच्चा लोग. कहाँ गए? अरे पागोटा बढ़िया आईची, हल्का हल्का वर्षा पकोची, निद आसुथिबो, I understand, but we'll have to look into the पढ़ाई also, है ना? तो फटाफट आ जाओ. Let's begin. Okay, the first tool is the enzymes. Fine. Enzymes मानव भितरे सबुठु crucial है restriction enzymes or molecular scissors. Restriction enzymes or molecular scissors. Now, scissors रो कामो कौन करे? Activa. ଆଉ ଖାଲି କାଟିବା ନୁହେଁ, ଠିକ ଜାଗାରେ କାଟିବା. At a particular site, desired site, ଯୋଉଠି ଆମ ଦରକାର ସେଇଠି କାଟିବା. Okay, so here restriction enzymes are considered as molecular scissors. Fine. Okay, these are the enzymes which cut DNA at specific sites. माने तमे यदि गोटे एंजाइम यूज करिवो, गोटे पर्टिकुलर सीक्वेंस रे ही से काटिवो. That is the specificity of restriction enzymes. जो जगह रे काटिवो ताकू हमें कहबा restriction site. जो जगह रे काटिवो ताकू कहा जाता है restriction site. And that enzyme is called restriction enzyme. ठीक है? Fine. And these enzymes, since they are digesting or breaking the nucleic acid, सेथिपाई की ताको कुआ गला कौन? Nuclease. Okay, so nucleases are enzymes that digest the nucleic acids, माने DNA और RNA. हमें एठी specifically कहा कथा कहूचे? We are talking about DNA. ठीक है? So basically restriction enzymes are digestive or hydrolytic enzymes that digest the DNA at specific sites, ठीक है, and form fragments. Say sites गुड़ी को क्या बोला? Restriction site. As you can see in this figure, एटा धरो तुम्हारा DNA है ला, ठीक है? Enzyme we are using is EcoR1. Fine. आंको details हमें आप और देखिवा. But एटी गोटे example से हमको just दिखाऊची. एटा है ला तुम्हारा DNA रो sequence. 5 prime to 3 prime, 3 prime to 5 prime. It has a sequence C, GAATTC, GAATTC. ठीक है? अच्छा, unique thing about this particular sequence is it has a palindromic sequence. को प्रकार रो sequence रे बाबू? Palindromic sequence. अरे palindromic क्या होता है? आगे से पढ़ो, from left to right, ये right to left, it is the same. देखो ध्यान से, एपटू GAATTC, तार next sequence रे देखो again this way as well as this way. The combination is same, isn't it? ସେଇଥିପାଇଁ ଏ ସାଇଡ କୁ କୁହାଗଲା palindromic side. ଯେମିତି for example Malayalam. If you would have heard the word Malayalam, Malayalam. एमिति पढ़िले भी मलयालम हबो, एमिति पढ़ले भी मलयालम हबो. देखो. So this is a palindrome. Fine. ओडिया रे कटक. Again palindrome. ठीक है. So any particular word or sequence, if read from left to right or right to left, forms the same. Therefore, it is called palindrome. ठीक अछि? बुझेला? Okay. So here, the restriction enzyme is cutting this particular site at this point. At this point, between G and A. Between G and A. देखो 5 prime to 3 prime रे भी G रू A भितरे काटुची and 3 prime to 5 prime with मतलब same G और A मध्य रे काटुची. So when it is cleaving the two sides, you will get this as well as this. ठीक है. एई जिनसे गुड़ी को कुआ जाता है sticky ends. What are sticky ends? केमिति काटला? That we'll see later. ठीक है. यहां तक समझ में आया? Okay. चलो आगे बढ़ो. Fine. Now in 1963, basically in 1963, ठीक है, two enzymes responsible for restricting growth of bacteriophages in E. coli were isolated. Now, question पूछा जाए कि why restriction enzymes are called restriction enzymes? A word restriction का use है. That is the thing which is being explained here. First of all, सेटा गोटे particular site रे काटुची. So the cutting is restricted to a particular site only. That is one reason. Secondly, these enzymes, they destroy bacteriophages. Bacteriophages रो DNA टाको काटी देवे. काटी देला परे bacteriophage रो DNA inactive हे जीवो. आउ bacteria को affect करि परिबोनी. As a result of which bacteria E. coli मध्य बंची गला. So since it is restricting the growth of bacteriophages, therefore it is also referred to as restriction enzyme. अरे बुझी पालो. Simple. ठीक है. Restricting growth of bacteriophages in E. coli. Okay. They were isolated. One enzyme added methyl groups to DNA. One enzyme added methyl group to DNA. कहार DNA E. coli रो DNA रे methyl group add कर देला. तो कौन है गला? से restriction enzyme आउ निजर DNA को destroy करिवो नाही. कहीं चिन्ही परिवोनी, identify करि परिवोनी. Okay. The other restriction endonuclease cuts the DNA. को DNA bacteriophage रो DNA. तो दीटा method रे एटी protection हैला. कौन? गोटे केस रे मिथाइल ग्रुप ऐड करदेला इन दी E. coli DNA. आउ गोटे मेथड रे कोन कला बैक्टीरियोफेज DNA को काटी देला. ठीक अछि. ना सेम सीक्वेंस ता बैक्टीरियल DNA रे मध्य थिबो, किंतु से बैक्टीरिया रो DNA को काटिबो नाही. Why? Because already methyl group added it. Since the methyl group is added, so the restriction enzyme will not be able to identify and hence कट करिबो नाही, कोनोसी डैमेज हबो नाही. बट वायरल DNA रे जेहेतु मिथाइल ग्रुप नाही तो सेटा को काटी देला. Hence destroying the viral DNA. ठीक अछि. And destroy हे गला परे बैक्टीरियोफेज रे से DNA टा आ कोनोसी कामरो नथियो. Understood? So that will restrict the growth of bacteriophage in the E. coli and protect the E. coli from damage. Clear? More than 900 restriction enzymes have been isolated from over 230 strains of bacteria. Bujela, naturally occurring ethical restriction enzymes ampagarebe ochi. Although sabuja ko amaro use hueni, but we have them. Okay? Oh. Next, look into the nomenclature. केमिति आमे नाटा देइथाउ कोनोसी restriction enzyme रो. First example that we saw was eco R1. ठीक अछि. Now eco R1 रे E stands for the genus name. Genus. ठीक है. Genus का मतलब है कहाँ genus रे? E. coli रो genus. That is Escherichia. E stands for Escherichia. ठीक है. ये जो CO stands for species, coli. R stands for strain, that is rough. And this is 1. 1 stands for the sequence. Roman number, the order in which the enzymes were isolated from the vector state. So first alphabet that is in capital, it refers to the genus. The next two refer to the species. R refers to the strain, co-type, and 1, so that is Roman numeral 1. It is the sequence co-order रे discover हेतला. प्रथम जेतु discover हेतला ए sequence रे इसका नाम दे दिया गया eco R1. ठीक है, देखो इधर. First letter indicates genus. Second two letters indicate the species. ठीक है. Eco R1 comes from E. coli RY13. This is the specimen from which it has been extracted. ठीक है. Is this clear, people? Restriction enzymes को यदि हमें classify करिबा, we have two types of restriction enzymes. One is exonuclease, the other is endonuclease. ठीक है. Exonuclease रो काम होला, they will cut the enzyme only at its ends. Only at the ends. बुझेला? They remove nucleotides from the ends of the DNA. माने 5' end row by 3' end row, they will cut the DNA. This is how the exonuclease works. Exo माने on the outside. So जो nucleus थी बाहर पटे कटला वो हो गया exonuclease. Then comes endonuclease. They cut at specific positions within the DNA. जेहेतु see हमें restriction enzymes रे exo, endo दुईटी जाको जिनसे use करता हूँ. But more importantly, the endonuclease are more helpful in the biotechnology. Exonuclease गुड़िकर use कम होता है, but they are also useful in certain cases. But majorly कहाँ को use करीबा? Endonuclease को use करो. ठीक है? Okay. They cut at specific position within DNA, bind to specific sequences or recognition sites, bind to specific recognition sites or sequences of the DNA and cut the two strands at the specific points. ठीक है. जेमती तमे eco R1 दिखतिला, GA TTC थिला, between G and A cut कला at both places. Result कौन हला? एपटे देखो गोटे sequence बाहरला empty, एपटे आउ गोटे sequence बाहरला empty. ठीक है. Fine. Since it is cutting between G and A, so A part टा अलगा है गला. A पर्यंत and A part टा अलगा है गला. Two parts are now different. See here. ऐसा, ठीक है फाइन। सो ए जो कटटा हैला एटी थाइमाइन एडेनाइन रो हाइड्रोजन बॉन्ड्स एक्सपोज अछंती, ठीक अछि। एठी मध्य एडेनाइन थाइमाइन रो हाइड्रोजन बॉन्ड्स एक्सपोज अछंती। These exposed hydrogen bonds are referred to as sticky ends. The exposed hydrogen bonds are known as sticky ends. Okay? And because of these sticky ends, they are able to join each other. Lagi pariba uthre puni. ठीक अछि? So जोटी से same sequence टी मिलियो तांको complementary rule हिसाब रे they will join. Clear? Understood? So ଏଇଭଳି ତମର endonuclease, exonuclease are used. Now, certain examples and important aspects about restriction enzymes देखो एटी. First restriction endonuclease was hind 2. First restriction endonuclease was hind 2. एटी मन रखियो था, आपके दिमाग में क्वेश्चन आएगा सर hind 1 कहीं कहलानी, why not hind 1? हमें कहूचे that 1 2 3 तो तांकर order, yes, तो hind 2 टा कहीं first चला, hind 1 हवा कथा, obvious कथा. Simple thing is initially जेतने ए enzyme restriction enzyme रे discovery हैला तो it was only known as hind, ठीक है, and its properties were defined, but तार किछि दिन परे again another category of hind was found. तो जेहेतु that was more basic to this, उसको hind 1 बोला, किंतु प्रथमे डिस्कवर किए हितला एटा हितला, सेति में आकु ना दे दिया गला hind 2, that was referred to as hind 1, ठीक है. Now see this hind 2, this restriction endonuclease comes from Haemophilus influenzae. That's why Haemophilus influenzae strain is D and sequence is 2. ठीक है, बुझेला? Okay. It cuts DNA by recognizing a specific sequence of 6 base pairs. This is called the recognition sequence for hind 2. कौनसी प्रकार रो restriction endonuclease तमे देखिबो, everywhere you'll find that they have, yes, 6 recognition base pairs. So say 6 base pairs को मिसे की तमरो restriction site व recognition site हेता है. Everywhere it is common. See here. ठीक है, recognition sites of various types of restriction endonuclease. Here ALU1, HAE3, BAMH1, hind 3, EcoR1, all are restriction endonucleases, ठीक है. लेकिन ALU1 and HAE3, ठीक है, they provide blunt ends, whereas BAMH1, hind 3 and EcoR1 produce sticky ends. So AE duity jinso, AE duity, they become exonucleases, ALU1 and HAE3, whereas these three are endonucleases. बुझेला? Okay. तो ये blunt end and sticky end माने कौन? Blunt end माने जोटी हाइड्रोजन बॉन्ड्स तमरा एक्सपोज हो नाहंती वो blunt ends है, जोटी हाइड्रोजन बॉन्ड्स एक्सपोज हो चंती वो sticky ends है. Simple concept. Hydrogen bonds exposed is sticky ends. Hydrogen bonds not exposed is blunt ends. Clear? Okay. Now look into this. Restriction endonucleases recognize a specific palindromic nucleotide sequence. Already I have told you this. Mane left to right, right to left, same sequence follow होती हो. That is called palindromic. ठीक है? It's a sequence of base pairs that read the same on two strands, 5 prime to 3 prime and 3 prime to 5 prime. ठीक है? जैसे यहाँ देख लो GA TTC CTT AG एटा तमरो इको R1 रे केमिति होउची एई पॉइंट रे से रिकॉग्नाइज करी तो काटुची. Okay, but a sequence थिले ही it will cut, otherwise it will not cut. Fine. So therefore you can see this is considered a palindromic site. Left to right, right to left, theta sequence 5 prime to 3 prime ho, 3 prime to 5 prime ho, the sequence remains the same. That's why it is palindromic in nature. Okay? Is that clear? Okay. Now, restriction enzymes cut the strand a little away from the center of palindrome. Little away. Kahe ki little away? So that there is no damage caused to the actual sequence. तमरो जो बड़ सीक्वेंस टी थिबो तार किछि टू मेन सीक्वेंस टू किछिटा दूर रे से माने काटिबे. Why? Because doing that it will keep the actual sequence intact. ठीक है? माने तादर कोनो डैमेज हबो नाही. Restriction enzymes cut the strand a little away from the center of palindrome sites, but between the same two bases on the opposite strands. Again, देखो ये बोलो और बुझो. Restriction enzymes cut the strand. This is very important. ठीक है? एक question पढ़तला exam रे. Restriction enzymes cut the strand a little away from the center of palindrome sites. Center थारू ଟିକେ ଦୂରରେ କାଟିବେ. But between the same two bases on the opposite strands. FT ମଧ୍ୟ G ଆଉ A ମଝିରେ କଟାଗଲା. In 5 prime to 3 prime रे 3 prime to 5 prime रे ମଧ୍ୟ same G ଆଉ A मझिरे काटिबे. ठीक है? They are called sticky ends. This leaves single stranded overhanging stretches at the ends. काहेकि अरे देखो 80 hydrogen bonds गुड़िको expose हेले. Here hydrogen bonds are exposed. Hydrogen bonds exposed. 80 hydrogen bonds exposed. जेहेतु hydrogen bonds are exposed, we call these sticky ends. Clear? And hydrogen bonds ro subidha ala, it will help in attachment of the sequence easily to any other similar sequence. जोटा कि तुम्हारा complementary थी वो. ठीक है? Understood? How? जेमती काटिवा पाई कि हमें एंडोन्यूक्लियस एक्सोन्यूक्लियस यूज करूचे. सेमती जोड़िया पाई कि मध्य हमें यूज करूचे enzyme. Now here you'll find the enzyme used to join the two sequences is called DNA ligase. ठीक है? DNA ligase. Just a minute. अरे बच्चों किधर गए? कहाँ गए रे बच्चा पार्टी है? इतने important class रे पिला नहांती. This is a very sad thing actually. Now ये चीज आपकी के लिए हो रही है. इटा तमो मनो पाई करा जाऊची. अगर आप ही इंटरेस्ट नहीं लेंगे, आप ही प्रेजेंट नहीं होंगे, फिर प्रॉब्लम है. Why? Because see, audio visual method रे पढ़ेवाटा is a very difficult thing as well as useful thing. Why? Because आप यही चीजें if you not learn it here, then in your respective institutes or colleges or schools वगैरह, they would be teaching you from the book itself. तो तमें से बही देखिबो, बही जहाँ बुझिबो, etc. But here you can actually see the things in a proper view. how they actually happen. Okay, and this will act as a better interface and you will learn more. Hmm. So they are present ରହିବା କଥା, but it is very disappointing as well as sorrowful कि people are not there. No, if it's a very frank thing. If in my days I would have come hold of such kind of a program, then I would have made the full use of it. But यहाँ पे बच्चों को मिल रहा है, they are not interested in taking it. This is sad. Anyways, ठीक है. How? So I insist on all of you. Suppose your friends are also there who are a part of this program. So please ask them to not miss any classes. Let that be physics, chemistry, maths, biology, whatever. Okay? Oh. Now, these sticky ends form hydrogen bonds. Already told you. Exposed कौन अच्छी ना hydrogen bonds अच्छी. Okay? These hydrogen bonds with their complementary cut counterparts. Okay. So sticky ends form hydrogen bonds with their complementary cut counterparts. This stickiness facilitates action of the DNA ligase. So DNA ligase static कौन कला ना फेविक्विक भलिया जोड़ी पकेला काकू those cut ends fragments. ठीक अछि. केमी join कला रे बाबू by simply creating hydrogen bonds. Creating hydrogen bonds. Hydrogen bonds thile se hydrogen bonds mano ko jodi pakaila. As a result of which पूर्ण sequence same megala. When cut by the same restriction enzyme, the DNA fragments have the same kind of sticky ends. Hence, they can be easily joined by DNA ligases. Is that clear? Bujela? तो हमें कौन जान ले मॉलिक्यूलर सीजर्स बा रेस्ट्रिक्शन एंजाइम्स बा न्यूक्लियसेस दैट इज एंडो और एक्सोन्यूक्लियस दे कट द स्ट्रैंड्स इनटू टू फ्रैगमेंट्स ठीक है exposing their hydrogen bonds. These hydrogen bonds are later joined by the DNA ligases. Yes. ठीक है as you can see here how the process is being carried out देखो जरा ध्यान से एटा तुम्हारा foreign DNA this is your foreign DNA fine आ भीतर कौन अछि तुम्हारा gene of interest अछि the desired gene तो तमे कौन करलो ना एठी यूज करलो endonuclease ना endonuclease तमे दीटा जगह यूज करिवो एठी मध्ये यूज करिवो to extract that particular sequence एठी मध्ये यूज करिवो to cut that particular sequence on the vector DNA बा plasmid ठीक है but गोटिए enzyme यूज करियो काहे की because the recognition site is same एठी जो जगह रे काटिबो सेठी मध्ये सेम सीक्वेंस रे काटिबो ठीक है मतलब मान लो वो जो आपने अभी थोड़ी देर पहले देखा था GAATTC is equal R1 ठीक है एंडोन्यूक्लियस तो ताको इधर एठी पकेली ये सही सीक्वेंस रे काटी पकेला GAATTC एठी पकेला तळे हियर आल्सो सेम सीक्वेंस इज देयर वो उसी सीक्वेंस में ही काटेगा GAATTC समझ में आया देन वंस इट इज डन हमें कौन करना ए जो फ्रैगमेंट ही बाहर ची ए फ्रैगमेंट ही आने की एठी जोड़िया ठीक है इस प्लाज्मिड में जोड़ेंगे हाउ डू यू डू दैट simply by using ligase now ligase जो बाहर ला हमारा using that ligase हमें a particular gene sequence बा कट फ्रैगमेंट को आने की जोड़ी देले कोठी into this plasmid. Why? Because through here this plasmid will enter into its host. This plasmid will enter into its host. देखि पारुथिबो तो एई जो जिंसटी तयारी हला एथरे कोन अछि एई पार्ट रे देखो मार्क करिवो this part contains the alien DNA alien माने जोटा बाहरु आछि ठीक है that is your gene of interest. Fine. So when the original plasmid is there, DNA, ताधरे हमें गोटे नुआ DNA strand add कर देले. So this is referred to as recombinant DNA. Recombinant DNA. ठीक है? Because of this recombinant DNA, transformation है गला. कार equalize रो. ठीक है? And then the host would become cloning. ठीक है? And the cells will divide to form multiple ones. सबुतरे कौन रहिबो original DNA रहिला along with the new fragment added to it. So multiple copies of the same particular recombinant DNA molecule will be generated. Same molecular DNA molecule will be generated. ठीक है? Hence this mechanism is very, very useful. Is that clear? ठीक है तो 80 restriction enzymes is over. ठीक है थोड़े restriction enzymes को भलो बार देखी दियो. Take a 1 minute gap. Just go through the NCERT where restriction enzymes are mentioned. Fine. And then we'll look into the vectors. ठीक है जल्दी जल्दी फटाफट